So Polyomaviridae, think SV40 when you see this. So SV stands for simian virus 40. It's a virus that infects monkey cells. Polyoma, the etymology of it is poly meaning many, oma meaning tumor, and viridae meaning virus. So when scientists first discovered this, they were actually looking for the causative agent of polio. But they also found Polyomaviridae when you're trying to isolate all the different viruses, and they found that this specific family of viruses have a tendency to produce tumors in different types of tissues. So poly from many tumors in many different tissue types. Some characteristics. So this is the first of the DNA viruses that we'll cover. It's double-stranded DNA, circular, icosahedral virus, naked. It's also small, so it's 45 nanometers in size, and its genome is 5,243 base pairs. And that's 72 capsomeres. And it's 3 capsid proteins. VP1, VP2, VP3. It has 4 histone molecules. And that is H2A, H2B, H3, H4. Okay, so looking at the characteristics, what stands out to me the most is the fact that it's a DNA virus and it has 4 histone molecules. And this illustrates the fact that, in essence, you can think of Polyomaviridae as a miniature chromosome. So just like how we thought a poliovirus as a miniature mRNA, here you can think of Polyomaviridae or SV40 as a miniature chromosome. And so you'll see that it actually uses it during its multiplication. It exploits a lot of the cellular machinery, and it has a lot of analogous genes, meaning that it has genes that do the same function as many of the cellular genes do in terms of regulating DNA synthesis and cell growth and so forth. And you see that because this thing acts as sort of like a miniature chromosome, you also see you can kind of see how it can also cause tumors in eukaryotic cells. So yeah, just think of this as a miniature chromosome. These histone molecules are the same histone molecules that our cells would use. Now, so I'm going to go to DNA characteristics now. We said before, it's double-stranded DNA. It's circular, has histones, super helical, and if you isolate the genome, it's infectious, right? Because essentially it's just a miniature chromosome. What happens if you put a miniature chromosome to eukaryotic cell? That chromosome can be replicated, it can be I mean, it takes a few more steps than that, but essentially it can be replicated, it can be transcribed. Right, and this miniature chromosome has genes. It has genes that literally push the cell cycle to start growing and to start growing and synthesizing DNA. So you take SV40 and put into a cell that normally doesn't usually replicate, then you can really turn that cell. The SV40 will force that cell to start synthesizing DNA and so forth. Okay, so now Polyomaviridae is normally it has two forms of DNA. Form 1 and form 2. Now, form 1 looks like this. Normal superhelical. Form 2 looks like that. So write this down. Take note of this because we're going to go into how you actually how you can actually isolate out SV40. So I'm just going to erase this tiny portion right here. On the side, I'm going to redraw that picture. 1, 2. So in order to extract out this virus, first you have your SV40 in your cells. Then you lyse the infected cells. Extract out the virus. And then you add in salt, basically salt water, 1 molar of it. 1 molar concentration of salt water, 4 degrees Celsius. What will happen is the DNA, the cell DNA, will be in a pellet, so centrifuge it. And the viral DNA will be in the supernatant. And then you do equilibrium centrifugation. In amphibian bromide. Now, when you do this, you can then isolate it according to form 1. So I'll draw the picture for it. You can actually isolate it according to different forms. And the reason for that is, let's say that this is mostly helpful in the laboratory, not really, as far as I can tell, not really in a normal setting, besides a normal setting in the laboratory. So if you're in a laboratory and you're trying to study this virus, one of the things you might need to do is to, loosen up the DNA. And when you loosen up the DNA, you want to get the loosened up DNA, the loosened up DNA. You need to separate that from the non-loosened form, like the unloosed, the tight form, the superhelical form of the SV40. And so the way you do that is you equilibrium centrifuge in amphibian bromide. And the superhelical form does not bind to amphibian bromide very well because it's in a superhelical form, so there's not too many spaces for amphibian bromide to bind anywhere. because there's little aetherium bromide binding, its density is high. So you might think, wait, but if there's less aetherium bromide, how can density be higher? Well, it's higher because then its raw density, like the original density of the virus, isn't polluted by something that's less dense. Here, you just have the exact opposite. High aetherium bromide, lower density. Yes, okay. So now we're going to talk about should be bullet point number 2. Okay. So now we're going to talk about SV40 and permissive versus non-permissive cells. First we'll go over permissive. So in a permissive cell, SV40 will have infectious progeny. So yeah, productive infection essentially. And you have lysis of cells. So think SV40, which is a monkey virus infecting monkeys. Right? It's permissive because the monkey cell permits the virus to enter it and ruin it. In contrast, you have non-permissive cells. Here, you have non-infectious progeny. The infection is not productive, it doesn't make any it doesn't make new viruses, and it instead transforms the cell. So you can't think of taking SV40 and infecting rodents. So this is important because it illustrates an important point about virus, and in particular Polyomaviridae, in that in order for the virus to do what it wants to do, which is to make more copies of itself, it needs to be able to infect cells of its host range. Only SV40 is only able to make more copies of itself if it infects monkeys. If it infects something that's not a monkey though, then it will cause tumor cells in that non-permissive host. So if you're in a lab or something and you have some animals that have a tumor and then you isolate a tumor and you filter and you're able to filter out an infectious particle, you can probably give a good guess that the virus that's causing tumors doesn't quote unquote belong in that host, meaning that it's infecting a non-permissive cell, and so then you get transformation, you get tumors. This also illustrates another point in that Polyomaviridae, when it infects a permissive cell, the viral DNA is free. And when we say free, it means that it's free to, it's just flowing around, chilling, replicating itself and everything. But if it's in a non-permissive host, then it's then it isogenizes, it gets bound to the host DNA. Alright, finally, we can finally move to multiplication. So the general steps, as always, is attach, penetrate, uncode, transcribe, replicate. So the attachment is in the attachment phase. The virus sticks to MHC1 via VP1. So that's the attachment. Penetration into the virus, into the cell. We have caveolin, receptor mediated endocytosis. For the encoding, virus loses BP2 and BP3. There's a protein called important. And it's important in encoding. So just think it's kind of like a wordplay. Just kind of think important, and then encoding. So about the important, the virus cannot uncode when it's in the cell. What is that? Okay, now we are going to move on to step 4, which is transcription. So here I'm going to start off with drawing a picture. You have to viral DNA. And you have early genes. I'll use that in green. And you have late genes. Basically, your termination site. Yeah. Now, when SV40 is transcribing, not replication, when it's in the transcription phase, the virus will transcribe this early side, quote unquote early, it'll transcribe these early genes before late genes. And the reason for that is because on the early side codes for T antigens. On the late side codes for VP1, VP2, VP3. Now T antigens tell the cell to grow. And when we say that a cell grows, there will be DNA synthesis and protein synthesis. Okay. Obviously, VP1, VP2, VP3 are capsid proteins. So it would make sense to make the T antigens first rather than the capsid proteins first. Why? Because you need to replicate your DNA, you need to make large amounts of protein in as little time as possible. So you'd want to make the protein factors that would tell the cell to do that, right? If you haven't even replicated your DNA, what's the whole point of making capsid proteins early? That's not going to help. In fact, it might actually be dangerous too, because if you make a bunch of viral proteins, it might alert the cell to, it might elicit even stronger immune response when the cell sees that, that there's now a bunch of viral protein inside of it. So this virus wants to do the T antigens first to trick the cell that everything is okay, or in the sense that everything is okay for DNA synthesis to occur and protein synthesis to occur. And then once it's able to replicate its genome, like many thousands of times, then it can quickly make all these viral protein capsid proteins, and then it can quickly assemble and get out. So that's the strategy behind doing this, and this strategy is called it's called some textbook. Divergent transcription. I keep thinking differential transcription. It's bidirectional. Okay. So that's transcription and translation for a slice. And then replication is just replication. So not too many details on that. You just simply replicate your DNA and so forth. Oh, by the way, all of this, except for protein synthesis, the transcription and the replication happens in the nucleus, just like Polyomaviridae. So just like flu, this stuff, the transcription and replication occurs in the nucleus of the cell. So moving on. Actually, I will give a recap of all switch color here. Let's switch colors. Let's use green. So you have your large T. Your large T antigens. So remember, T antigens are basically viral proteins. They're just not the capsid proteins. The T antigens are the ones that tell the cell to go make DNA and translate proteins and so forth. There are 4 T antigens: large, middle, small, and tiny. So large T antigen does a few things. Assembles DNA synthesis machinery. Unblinds viral DNA. It binds to retinoblastin to activate E2F and increase cell growth. And of course, when you increase the cell growth, it means to make DNA and make proteins, and specifically make the viral DNA and viral proteins. It also binds to J. It also binds the J domain to increase cell growth. Binds P53 to stop apoptosis. It binds P300 to stimulate to binds P300 for some genes and repress others. So large T antigen does a lot of stuff. You can see here some of DNA synthesis machinery, perfect for viral DNA synthesis. It needs to unwind the viral DNA so that the viral DNA can be copied and can be transcribed and copied later. Now, it binds to RB to retinoblastin so that it can liberate E2F so that it can increase cell growth. So these proteins right here is RB, E2F, J domain, P53, P300. These are all in the cell. These are cell proteins. And essentially what these cell proteins do is they tell the cell to stop growing and dividing, to stop DNA synthesis. Right? You don't want, for example, you wouldn't want the cells in your retina or of your eyeball to continue growing. Then you won't have a working eye. So you have these things there, these genes there. It's RB, E2F, J domain, P53, P300 to inhibit cell growth. But SV40 doesn't want that. It wants to be able to multiply and proliferate. So the way it does that is that it takes T antigen and binds to these genes that stop cell growth. And in doing so, it tells the cell to start DNA synthesis, to start translating proteins. RB is actually a negative inhibitor of E2F. So E2F is a cell protein that tells the cell to grow. But its E2F naturally is bound by RB, so it doesn't do, so E2F can't do its thing. But if you take away RB by using T antigen, now E2F can go wild. J domain is native and it tells the cell to not grow, so you bind it. with T antigen, you can get the cell to grow. So just P53. So P53, this one tells the cell to kill itself. And the reason why it does that is that it's a defense against viral infections, actually. So when you infect a cell with SV40 or just any virus, P53 levels will shoot up. And that's so that the cell can kill, a virally infected cell can kill itself to stop the virus multiplication dead in its tracks. So in order to counteract this, SV40 makes T antigen to block P53. And if you block P53, then you don't get any more apoptosis. You don't have any more cell death, then your virus cannot replicate. B300 just express some genes and repress others. You want the right genes expressed at the right time. And this is for viral genes. Okay, small T antigen. This one binds protein kinase 2A to drive cell into S phase. So there's actually a good picture in a textbook for that system. I'm going to draw it out here. So normally, you have P-phosphoprotein kinase 2A inhibiting this protein called MAP kinase. Now, MAP kinase without PP2A, it will get phosphorylated and you get transcription, you get cell growth, and synthesis of cellular RNA. So in order to control that, the cell has something called PP2A to bind to the MAP kinase, so that you don't get unnecessary RNA transcription. What SV40 does is that it takes a small T antigen to prevent the PP2A from stopping MAP kinase. So essentially, small T antigen frees MAP kinase to do what it will do. The end result is you drive the cell into synthesis phase. diversity antigen. And it tells the cell to proliferate and divide. Find your tiny t antigen is no known function. Tiny t antigen. Okay. I'm not gonna write this down, but VP1, VP2, VP3, these are capsid proteins. Okay, moving on. RNA synthesis happens in your nucleus. Your cell RNA, as before, it exploits the cellular machinery for RNA synthesis to synthesize viral RNA transcripts. The virus also exploits cells transcription factors. And these include the TATA binding protein, and a TATA binding protein is a part of TF2D. So let's illustrate this. Here's your TATA box. Okay, so this is the viral genome. It has a TATA box, and basically the TATA binding protein will bind to the TATA box, and from there you just basically get transcription. So I'll do this a bit. I'll do my best too. So data is bound. Now this is a triplet of 21 base pair repeats. Base pair repeats. And the repeats are lysine and cytosine. And this protein, called specialized protein 1, abbreviated as P1, binds here. Your enhancer increases transcription. Basically, the virus uses our cells do very similar thing. These are cell proteins, and yet the virus DNA has the stuff that these cell proteins would attach to. And so what it's basically telling the cell to do is tricking the cell, lying to the cell, and saying that, oh, I'm a viral DNA, or oh, I'm a cell DNA, technically I'm a virus DNA, but you don't know that because I have these right here. So bind to here and transcribe us. Pretty deviant. We want to copy that down. I have a few more things right over here. So some other things to know. It's very hard. The virus splices RNA transcripts. To get the different T antigens. Okay, so lizard, large T antigen, and your small t is just this. Okay, basic picture. The large T antigen is the same sequence of DNA, but for large T antigen, the virus would splice this middle portion to get the large T antigen. So get these two together. The small T antigen just cut this whole upper 2/3 and get this. It's not actually 2/3, but you get the point. Cuts away this entire region, and only saves this last part.